In this video, we will discuss about how glycans can be used to modulate the immune response. So, in the normal immune response, what happens is that these antigen-presenting cells or the matured macrophage, which is also known as dendritic cells, that it has the information about non-self epitopes that are present within the host, and this antigen-presenting cells will provide this non-self epitope information to these naive CD cells, and thereby activating these T cells, and these T cells then act against those non-self epitope containing uh, cells that we can see here. This is the various ways by which this T cells can activate. T cells can T cells gets activated, such as this the release of interleukin 10 and this TGF beta cytokines. These regulatory T cells will get activated, and the other T cells functions are also shown here. A main topic of discussion in this video is about this glycans that also plays a role in providing the non-self epitope information to these antigen presenting cells. And one of the key receptors that helps in detecting the glycan structure is this C-type lectin receptors. It detects the glycans that are present on uh, the viruses or on the cells that are harmful to the host. And and since glycans this glycans also plays a role in this antigen, in the determination of this antigen, so non-self antigen by the antigen presenting cells. There is a push to use this glycan. There is a push to use this glycan structure to train these antigen presenting cells about the non-self epitope that are present on the cancerous cells. As in the cancerous cells, it has been observed that there is aberrant and glycosylation that are observed, and there are different strategies that are been going on, where glycan is used as a intermediate to glycan is used as an intermediate, or uh, glycan is used as a we can say receptor to so that these antigen presenting cells will be able to determine will be able to differentiate between the harmful cells and the healthy cells by presenting the non-self epitope through this glycan into by providing the information about non-self epitope with the help of this glycan to this antigen presenting cells. And some of the examples that are being tried is shown here. It has been found that these high mannose glycan structures are being recognized by these C-type lectin receptors, thereby different different carriers are being modified with these high mannose structures that we can see here. These nanoparticles are being modified with high mannose carbohydrate structures, liposomes, polymers, glycostructures, protein and peptide conjugates are being modified these high mannose structures. Thereby they help in thereby the possibility of these liposomes or nanoparticles to be detected by this APC is increases, and thus the epitopes that are present within these liposomes or nanoparticles will be determined by these antigen presenting cells, and then it can activate the host immune response to act against those uh, non-self epitopes, which in uh, which in normal forms not been recognized by which in normal form are easily been evaded by immune surveillance because of the various factors such as tumor microenvironment and glycocalyx that we have covered in brief in our previous videos. And this is one of the examples that I have already discussed in one of our previous videos, but since it is related to this topic, I'm covering it again. That CAR T cells can be used. CAR T cells right now is been used to treat, is been efficient against the blood cancers, because in blood cancers, the CAR T cells are being modified to target against the cancerous blood cells by providing the antigen of these cancerous blood cells on this CAR T cells receptor or chimeric antigen receptors. However, this CAR, that is chimeric antigen receptor, can be modified with the aberrant glycans that are present on the tumor cells. Thereby, these CAR T cells can recognize these tumor cells, will be able to distinguish these tumor cells from the healthy cells and can act against those tumor cells easily. So this, in other terms, exact accelerated the immune response of the host by providing the antigen receptors directly to the immune cells uh, instead of waiting for the immune response, instead of waiting for the immune system of the host to determine this antigen that are present on the tumor cells. So this accelerated immune response will prevent the exaggerated condition of the cancers within the patients, and thereby helps in curing the patients without too much pain, or without going to the more detrimental and painful stage. And some of the antigens that are found on the tumorous cells that are mentioned here, such as the silyl TN, which are found in tumor cells in a higher amount compared to the healthy cells, and this Lewis YGT2 are the other glycans also. So I hope you have understood how these glycans can be used to modulate immune response. And if you need to understand more about this glycan modulation immune response topic, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.